think I will just continue. I cannot see anybody's comment here. Actually, the reason for the comment is to be sure that you guys got the right link. So if you have been and if I see at least one comment, then I will just proceed. I've started, you can see I'm now almost halfway. I can already discuss question 3 and what is question 4, 5, and 6. But I think the method I followed is not recording. So I cannot confirm whether you guys are actually with me. So I don't know how I can call your attention. It seems to already following the recorded part of this edition. Oh, good. I see someone now. Samu Lamu. Samu Lamu. So good day. But I think I should just continue. I've been waiting at least to see one person join, so that I'll let you know that there is a person following you. So if you have a question, Samu, while there's your question, then you can move together. But I think what I will do, I will have to go back to the from beginning. Like I said, I've gone halfway, but audio has not been following me. So the method I chose to follow by condemning the link to you. So I think I shared actually the wrong link, and then we ended up taking the recorded parts of which it wasn't even, it was an AI summary of the discussion. So now that you are here with me, what I will do is I will go back and start from the first question so that yeah, I can see Chama Chama is here also going. So I'm trying to this is okay. So here we are system. All right, so let me start from beginning. You're welcome to Brighton 241. First question, biochemistry of macromolecules. Um, as I promised you, as I promised you the other time, I planned actually to go through at least like 3 to 4 questions paper. Yeah, but unfortunately, I got the question paper at a point of preparing this short slides for the interaction. Unfortunately, I looked at those questions paper, I searched for them, but I didn't look at them. So I thought I just continue with the one I was able to lay my hands on. So I was only able to get the immediate first question on weekdays or weekday group. You understand what I'm talking about. So I will go through this question and then if you have any question, please do not hesitate to ask the question in the WhatsApp group. So during the discussion on the WhatsApp group, for those of us that we are not even part of us, they can later on follow the recorded access of the recording and then also follow the WhatsApp description. I am very much sure they will catch up with whatever being discussed here. So, like I said, what I put a highlight or rather an answer to those questions. An answer to those questions in short form, in short sentences. Because the truth is, these are very simple questions. These are very simple questions. They are questions like we always say that you can answer them in 2 or 1 sentence. Some of them are even in 1 sentence. You can put 1 sentence, 2 sentences, and a 3 sentence. Very few of them you can have up to 5 to 6 sentences. But yeah, sometimes students prepare to elaborate. Yeah, you can elaborate, but at least while elaborating, don't talk too much to the extent that you may lose the focus. Lose focus, sometimes students lose focus and ended up saying things that were not even what is expected of them to say, or rather to even contradict themselves. So, um, advisably, just to be short and precise, but it doesn't mean you shouldn't explain and number you shouldn't explain yourself, number one. Number two, it doesn't mean you must present it the way I presented the answers here. In fact, the way I presented the answers will be very difficult for you to present them in a very short form like this. So try and elaborate them, but elaborate them in your own language. I am sure if I am to ask you to write what I've the answer I drafted here to a particular question in your own language, I know it can be 1 sentence to me, but of course to you it might be up to 3 sentences. So that is well understood. You talk with, maybe you have a very long sentence or 2, 3 sentences explaining 1 point or 2 points. So that is well understood. So, but please avoid writing too many things. Just 1 question and you're consuming a whole page, a 2 page, for example, maybe 1 page or 2 page back and front is too much. Sometimes students talk too excessively on questions on things that were not expected to go elaborate. So, um, let me start with the first question that says, which is question one, says highlight how glycoproteins are involved in ABO blood group grouping. Remember while we were talking about cell membrane, we talked about the component of cell membrane. And the basic component we mentioned were the lipids and the proteins. We said cell membrane contain or is made up of phospholipids, and these phospholipids, in addition to the or other type of lipid like cholesterol that control the flexibility, that control the flexibility, and also prevents the membrane fluidity. And we also say the proteins. And these proteins, they are of two types. We have the peripheral membrane protein and we have the integral membrane protein. And we say in most cases you can have carbohydrate, but in carbohydrate when found on the membrane surface, they are either attached to these integral membrane proteins or attached to the lipids. When they attach to lipids, they are called glycolysis. When they attach to proteins, they are called glycoproteins. So in either case, either the glycolysis or the glycoproteins, APO blood grouping system is a system that is determined by either the glycoproteins, like I said, or the glycolipids. So in either case, you can have this glycoprotein or glycolipid being a determinant of the ABO blood glucose system. And how it happens is usually on this membrane lipid or membrane protein, you have a trisaccharide, a three molecule of sugars. These three molecules of sugars are glucose. Remember, we said glucose. We'll also mention N-acetyl glucosamine, and then we mention galactose. But they occur in this sequence. You have glucose, you have galactose, you have N-acetyl glucosamine. So these are the triglycerides that are found on the surface of red blood cell membrane, either attached to the lipid or attached to the proteins. These are the baseline triglycerides. These are referred as the antigen. So when the trisaccharide baseline only occur on the surface of the membrane of an individual or red blood cell membrane of an individual, that baseline is considered antigen O. So that a particular individual is having a blood group. But when you have some individual where on this trisaccharide, this glucose, galactose, and acetyl galactosamine, you have N-acetyl galactosamine attached to N-acetyl glucosamine. These individuals were said to have blood group. While there are also a set of individuals that have, instead of this N-acetyl galactosamine, they have galactose attached to this terminal uh, N-acetyl glucosamine instead of N-acetyl galactosamine as in E. So we have galactose attached to N-acetyl glucosamine that made those individuals to have blood group B. While there are individuals whose on the surface of their membrane, the red blood cell membrane, they have these trisaccharides, and these trisaccharides contain both the after the baseline trisaccharide, they also contain both those ones with N-acetyl galactosamine, while there are others with galactose. So those individuals were said to have uh, blood group EB. So this is just what you will explain that the blood group system of an individual is determined by the presence of the glycoproteins or glycolipids on the surface of the red blood cells. Usually, an individual with a baseline triglycerides, which comprises of glucose, galactose, and N-acetyl galactosamine, were referred to have O antigen or H antigen. Therefore, they have blood group O. While an individuals, in addition to the H antigen or the trisaccharide, they have N-acetyl galactosamine attached to the final or peripheral N-acetyl glucosamine of the trisaccharide to form antigen E. So those individuals were classified as those with blood group E. While you have another individuals that on the surface of or on the peripheral and acetylic glucosamine, they have galactose attached to it. And those individuals they are classified as those that have blood group B. while individuals that have both the two A and B were classified as A. A and B meaning you have one trisaccharide with N-alcyl galactosamine, while one trisaccharide and with galactose attached to their final N-alcyl glucosamine residue. So they have this one and they have the other one. So they are called, or they are regarded as individuals in A antigen and B antigen. Therefore, they are classified as individuals with blood group A. So basically, this is what you were expected to mention. So briefly, in one or two sentences, it will answer. Now, quickly moving to question 1B. Question 1B says define a buffer and explain how it regulates the pH of the solution. So it's one question that looks like a two question. Define a buffer. So you start by defining buffer. Buffer is a solution that resists a change in pH when small acid, a small amount of acid or a base added to it. You can go ahead and elaborate more by saying a buffer contains or is made up of two major components, which are the weak acid and the conjugate base. I think basically. So you can now go ahead. The next question says explain how it regulates the pH of a solution. So in explaining how it regulates the pH of a solution, one thing we know that yes, a buffer contains the conjugate acid and the conjugate base. Yeah. So how does these things work to maintain the pH, to reduce a change in the pH? For example, we studied when you add small amount of acid or you add a small amount of base, the pH still maintains. How is that possible? We know that an acid is a substance that contains a lot of hydrogen. Hydrogen. So basically, acid contains a lot of hydrogen ion, while the base contains a lot of hydroxyl ion. So if you add an acid to a solution of a particular pH, what will happen to the pH? Definitely the pH will go down. So what is expected is in a buffer, generally buffer does not immediately change the resist the change in the pH. Meaning if you add an acid, yes, of course, because the acid contains a high concentration of hydrogen ion, of course the pH will go down. So when the pH go down, so and when the pH goes down, of course, what happens is the pH, the conjugate base, will now react with this excess hydrogen ion so that it will undergo what we call neutralization reaction. So the excess hydrogen ion will be seeped out or converted to water. Therefore, until the initial pH of the buffer is being restored. Likewise, when you add a base, what happens is the acid will now react with this hydroxyl ion, and this reaction leads to formation of water. And when water is formed, it's reduced or neutralized the change of pH. Of course, when you add the base, we know that the pH will go up. So by the time the reaction between the weak acid and the buffer, the hydroxyl ion continues, then the pH will start coming down until it reaches the normal pH of the buffer. So once it reaches the normal pH of the buffer, it will not go up down further. So this is basically the mechanism. The mechanism involves the reaction between the weak acid and the hydroxyl ion, or the conjugate base and the hydrogen ion as being found in acid and base respectively. And remember, we said the acid contains a lot of hydrogen ion concentration, while the base is defined with the presence of high concentration of hydroxyl ion. So the hydroxyl ion will react with the acid to form neutralization reaction, and this will lead to removal of the excess hydrogen ion that comes in form of base. While in case of acid, the weak acid, the conjugate base will now react with the hydrogen ion that comes from the added acid until all the hydrogen ions were converted to water, then the pH will be maintained. So this is basically how the buffer maintains the pH. Now going to the next question, which this question. 1C says, state and define each term of Anderson-Hasselbalch equation. So this is basically something that looks like also two questions like the previous question, where I will say first of all, you state the Anderson-Hasselbalch equation, and then you define the terms in Anderson-Hasselbalch equation. So stating the Anderson-Hasselbalch equation, you just mention Anderson-Hasselbalch equation says O is pH equal to pKa plus log bracket concentration of uh, concentration of conjugate base all over the concentration of the acid. Simple. Put the terminology the way it is supposed to be and the way it was written here. Because some of you actually point out making a mistake of changing. Is it plus? Is it minus? All these kind of things. Or is it the conjugate base on top or the acids down? Something like that. Some people were just, during the graduation, you reach a point whereby the hydrogen ion moved, sorry, the Aussitz is down while the, the conjugate base is up. Initially, the acid is up for the conjugate bases. So some people were fond of making those kind of mistakes. So while Mike and I came across actually quite a number of this, and so during the graduation, you have to know, you have to understand the graduation, how the graduation occurs, and then you get the final formula you obtain. So now you actually know what is the final formula. So you define the terms. pH, pH is the measure of the acidity and alkalinity of the solution. pKa is the acid dissociation constant. In this case, it's the weak acid dissociation constant. And then you have A, which is the concentration of the conjugate base, and HA, which is the concentration of your weak acid. You define that in this way, or you can just say pH equal to measure of acidity and alkalinity of the solution. pKa equal to acid dissociation constant. Hydrogen concentration, A in this square bracket minus its conjugate base or concentration of conjugate base. HE in square bracket the concentration of weak acid equal to concentration of weak acid. Put it this way, I think if you did that, it's something like that. In 1 minute you can do that. Now moving to question 2. Question 2A says with the relevant example, briefly explain the following: aldose ketose isomerism. So if I were in your position, I would just define isomerism. What is an isomerism? I will know isomerism is a terminology used to describe the situation when you have molecules of the same molecular formula, but the different spatial arrangement, different structural arrangement. So in the case of aldose and ketose, we know that in aldose and ketose, specifically if you take a particular sugar, a 6-carbon sugar, you will discover that this 6-carbon sugar, if you are to write the molecular formula of this compounds, they are the same. But the structural formula and the spatial arrangement are different. These ones will have a ketose, while these ones will have the aldose. So the basic difference is the presence of this aldehyde group, while in the case of ketosis, we are having the ketone group. So this is basically, and you can remember any of these aldose or a ketose. You can just give an example of an aldose or a ketone. Now, moving to alpha and beta anomaly. This is the same question. First one is Roman people 1. This is Roman people 2. So this Roman people 2, you write a short note on the alpha and beta anomalies. So here you can also go ahead and tell us what is an anomaly. Remember what we discussed regarding anomaly. Of chiral carbon, chiral centers, where we say all the sugars, all the carbons were chiral carbons, with the exceptions of those carbons that are regarded as the functional carbons. So the carbonyl carbon and the ketone carbons were not anomeric carbons. But when we go down where we discuss cyclization, the cyclization reaction formation of furan and pyranose in Howard's production formula, we made mention that this cyclization reaction leads to change of this carbon from non-chiral to a chiral carbon. Formation of chiral carbon leads to group around this carbon, this functional carbon that was transformed now to anomeric carbon is what define the alpha and beta anomers, and we said that the OH group, when it is at the right or off, it is regarded as alpha, or when it is down, it's regarded as beta. So this is basically what you expected to mention. While in the case of D and L isomers, we talked about enantiomers, we talked about in enantiomers, we said we have a difference in arrangement, hydroxyl ion arrangement within a particular carbon, which we give examples in the case of mannose, galactose, and glucose at a particular given. So in D and L isomerism here specifically, so I will start by telling us what is enantiomers, because the D and L isomers are enantiomers. Enantiomers in the sense that you they have the difference is on this one carbon, which this carbon is the carbon adjacent to the primary alcohol, which is carbon 5, I think. So this carbon 5, you have the OH group oriented by the right, it's regarded as D, while if you have the OH group to the left, this is regarded as L. Now, question 2b. Question 2b says state two main constituents of stress. So this is a very simple direct question. You just mention, if you like, just say one, amylose, two, amylopectin. You don't have to mention any other thing. But if you like, the kind of person that like following, or you have always something to say, you can say the two main constituents of stress are one, amylose, two, amylopectin. That is it. And you got the score allocated to this question. As simple as that. Now, moving to question 3a. Question 3a says enumerate four cell organelles and their function. So, if I like, I will just mention, list the four, mention the four, and then talk about their functions. There are many. We talked about many of them, starting with the cell membrane, the cytosol, the nucleus. We talked about the ribosomes. We talked about endoplasmic reticulum. We talked about peroxisomes. We talked about lysosomes. We talked about mitochondria. We talked about endoplasmic reticulum, the two types, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. We talked about the Golgi apparatus. So you can mention just four and then talk about their function. This is a very simple and question that you might have even answered during your secondary school days. Now, question 3b says describe the chemical bond in a dinucleotide. I remember while talking about the chemistry of nucleic acid, where we introduce nucleotides and define what are nucleotides. We said that nucleotide contain two major components, and these components include the nitrogenous base, the phosphate, the ribose sugar, and the phosphate group. And we let mention that the nitrogenous base is attached to the ribose sugar. They attach to the ribose sugar through a type of bond we call glycosidic bond. And then we have a phosphate group attached to this nucleoside. So when you have the nitrogenous base attached to the ribose sugar through the glycosidic bond, we have a nucleoside. And then we have the phosphate group attached to this nucleoside to form what we call a nucleotide. And we say the phosphate group can be attached to either carbon 3 or carbon 5. So normally in DNA, you can see that the OH group is present at 3 carbon. You have at carbon 2, but carbon 2 undergo a reduction reaction where the OH group was removed because the sugar, the ribose sugar was reduced to the oxyribose sugar. So the OH group now left is only the OH group on carbon 3 and the OH group on carbon 5. So the OH group on carbon 3 and carbon 5, they have the potential to react with the phosphate group. So normally in nucleotide formation, we said the phosphate group is attached to carbon 5. That is why we have nucleoside 5 monophosphate. Nucleoside 5 monophosphate. All of them attached to carbon 5. So when you have a nucleotide with a phosphate group attached to carbon 5, and you have another nucleotide with a phosphate group attached to carbon 5, and these two need to come together to form a bond to form a dinucleotide. So what we said is the bond is formed between the, the carbon 5 and carbon 3. So the phosphate group that will attach to the carbon 5 of one of these nucleotides will now react with the OH group at carbon 3 of the other nucleotide. So this phosphate group will now, one of the phosphate group will now form a bridge between the two nucleotides. It will form a bridge. So you have the phosphate group initially attached to carbon 5, and that same phosphate group now attached to the OH group at carbon 3 of the other nucleotide. So you form a bridge between carbon 3 and carbon 5. That is why we call it 5 primer, 3 primer, 5 primer phosphodiester bond. That is actually where the names comes from. So this is just what you explain, and by explaining, you have your max allocated to the person. You got it. So it's not a big deal actually. Okay, I understand. So the next question, question 3C says, describe the maturation and renaturation of DNA. So remember, you can also remember this one uh, while discussing the structure of DNA. While discussing the structure of DNA, we said the DNA exists in double and a helical form. It doesn't exist just as the polynucleotide single strand. Rather, we said the two single strands have to hybridized to form double helical structure, where we said adenine will bind with thymine, while guanine will bind with cytosine. So if that happen, what happens is you form a double stranded molecule, double stranded molecule that wind cycle or coiled around a particular axis to form a helical structure. So this helical structure, we say, can undergo denaturation reaction. And the denaturation, and this denaturation is nothing but to lose in this double helical structure. And how does the lose occur? It's just the separation of the this hydrogen bond, the hybridization reaction, the hydrogen bond between the adenine and thymine, where we say adenine and thymine, there is two hydrogen bond, while guanine and cytosine, we have three hydrogen bond. So there are quite a number of factors that can instigate this denaturation. We mentioned is temperature, high temperature. The high temperature can actually break these bonds. Therefore, the two strands will break into a single strand. Another thing we said is the ionic strength. The ionic strength can also causes the dissociation or breakage of this hydrogen bonds. And another thing we said is the pH. The pH, either the acidic pH or the alkaline pH, the high acidic pH or high alkaline pH, can actually cause the disruption in this hydrogen bond that exists between adenine and thymine, as well as guanine and cytosine, causing the dissociation of the distinct interaction double helical structure interaction, leading to formation of a single or double strand, leading formation of a separate strand of DNA. So at the same time, when this condition goes, for example, maybe it's high temperature, and then you remove the, you lower the temperature to normal, they can rehybridize. They can come back again. So this process is what we call renaturation, the joining of the complementary strand when the condition is restored. And this is the explanation given, and yeah, I think you'll be able to say this short sentence, a very 2 or 3 sentences. Yeah, good to go. You've got the facts you expected to. And then the next question, question 4A, says enumerate the classes of lipoproteins and describe only one of them, just one of them. Describe one. So you can just list them. Chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. Sorry, I arranged them from the highest to the lowest with respect to the density. So you just pick one, any one, low density. So here I said low density, and I said low density contains 70% of cholesterol, and its main function is the transportation of cholesterol to tissue from the liver. to extrahepatic tissues. So, so you can say tissue liver is also a tissue. So extrahepatic meaning any other tissue beside or outside the liver. So that is the function of low density lipoprotein. So it's move the cholesterol to, and we said the excess amount of cholesterol, you know that in the extrahepatic is responsible in a number of cardiovascular diseases such as atherosclerosis. That is why when you have excess of this low density lipoprotein, it is considered as a danger, as a danger signal to cardiovascular disease, and it's why it is called bad cholesterol. So also you can decide to talk about high density lipoproteins, that is involving transportation of cholesterol to the hepatic cells where they convert it to bile and then bile salts and then excreted out. Or you can talk about chylomicrons being formed in the intestine and basically meant to transport the ingested lipids to different organs of the body. So you can just choose one and talk about it briefly. Then question 4b says, describe how essential fatty acid deficiency causes infertility. So one important thing you just have to remember while addressing this question is that essential fatty acids, remember while talking about the formation of prostaglandins, formation of steroid hormones, etc. We made mention that we started the discussion with fatty acids. We say some of these fatty acids are essential, while some are non-essential fatty acids. We said the non-essential ones are the ones that we can produce in our body, but the essential ones are those ones that we cannot produce. And we said those essential ones are being implicated in the synthesis of quite a number of molecules like prostaglandins and steroids. And these prostaglandins and steroids made mention of their basic roles as the signal mediators as well as the involved in the synthesis of hormones, most especially the steroid. So they're involved in the synthesis of some sex hormones like progesterone. So if you're not taking these essential fatty acids, causing you're going to have a risk of developing deficiency in synthesizing these sex hormones like progesterone, and you may fall a victim of infertility. So basically, this is what you're supposed to capture in your own language. I'm sure once you capture that, I'm sure that it will solve this language. So the next question, suppose he say describe the formation of peptide bond. In peptide bond, what we mentioned is that the two amino acid comes together to react to form a peptide, a dipeptide. So when a dipeptide is formed, it is formed between the carboxyl group of one amino acid and the amino group of another amino acid, where the carboxyl group of one amino acid and the amino group of one amino acid comes together to a process we call a condensation reaction. And this process leads to the release of water, where you have the OH group coming from the carboxyl group and the hydrogen from the amino group, leading to the release of water. This is what it is called a condensation reaction. So at the end, you have the carbon forming at the bottom with nitrogen. That is a classical forming a bond with nitrogen. That's a classical condensation reaction. This is how a bond is formed. Now the next question, question 5, says explain why triacylglycerol liberates more energy than carbohydrate and protein. So one of the most important things to mention here is that triacylglycerols contain a fatty acid, three fatty acids attached to each carbon in the glycerol. So you have carbon 1, carbon 2, carbon 3, all attached to it. It's only glycerol that has this OH, 3 OH group at attached to each carbon. So apart from that, then you only have the only oxygen present is on the carboxyl group of the fatty acids, the individual fatty acids that were attached. So you can see that fatty acid is less with respect to the number of oxygen atoms. So it's now considered to be less oxidized molecule. And we know that the process of energy production is the classical oxidation. Classical oxidation of the carbons, classical oxidation of these carbon chains is what release the energy. And now we said we have two molecules, one that is partially oxidized, another one that is less oxidized. And of course, if you are oxidizing the two, the extent of the oxidation will be higher in this one that is not oxidized, while the partially oxidized will only require a few oxidation steps to be completely oxidized. And this is the simple reason why the triacylglycerol glycerol contain high energy because it is highly reduced, while the carbohydrates and the proteins are they are highly oxidized, partially oxidized, so they require little steps of oxidation to be highly oxidized. Remember, even while defining carbohydrate, we say polyhydroxy ketones and polyhydroxyaldehyde, meaning they have a lot of hydroxyl group. For them to have a lot of hydroxyl group inside here, of course, they are highly oxidized molecules. So here you can even add that 1 gram of triacylglycerol gives 9 kilocalorie of energy, while 1 gram of carbohydrate only yield 4 calories of energy, which is more than 2 times. So the next question, 5B, says enumerate 4 techniques used in purification of protein. So we have precipitation process, salting out. Remember, we talked about using of ammonium sulfate and some other salts that can use in solubilization of or that reduce the solubilization of protein in the solution. And we've mentioned the mechanism how these ions interact with these atoms in a protein, thereby reducing the relationship between the water and the proteins. We talked about solubility of the proteins, and we talked about this property of water. Remember, in that property of water, I give a detailed explanation on where you can even explain how water interacts with substances, why the interaction is not possible. When you said the interaction is not possible, what made that interaction not possible? Chemically, you can be able to do that. If you don't, please take your time. I'll read through this our recorded class on that. I'm sure I'm a bit elaborate on that. And I've related how this precipitation, how does this salts used in precipitation has been, or how does it interfere with the interaction between the amino acid in the protein and then the what do you call it, the water molecules, thereby reducing the solubility of it, making it to precipitate out of the solution. And then we talked about separation also using dialysis, where we said you have the dialysis bath that contain a very small force where the proteins they cannot pass through, but the water molecules and the small other substances can pass through. That is separating the water molecules and the small molecules from the protein. We made a mention of chromatography when we separating the proteins on the basis of different properties between the on the basis of the size, where we mentioned that you can use gel filtration chromatography or size exclusion chromatography. You can use affinity chromatography on the basis of affinity between the different types of proteins. You can also use ion exchange chromatography, where we said you can separate on the basis of the change on these proteins. We also talked about electrophoresis, where you can separate on the basis of size as well as the charge of this things. So please, you can also think of other techniques. There are other techniques we mentioned, so you can add any one. Just what you expected is to mention four, but just to mention four. So generally, we talk about the kits or the fatty acid, for example, where you the hydrophobic region and the hydrophilic region. And the hydrophobic region being the carbon skeletal, and then the hydrophilic region being the carboxyl group, because the carboxyl group contains the oxygen, and this oxygen can easily interact with water. And then also, if it happens in the form of triacyl glycerol, we say the glycerol contains the hydroxyl group, you can easily interact with water. Likewise, also if it is in phospholipids, where you have the phosphate group is highly for that, you can easily interact with water as well. So this is basically what you expected to. And the particle nature of lipids is the fact that the lipid contains some components such as the phosphate group, such as the glycerol, such as the carboxyl group in fatty acid. So glycerol in triacyl glycerol, phosphate group in and the backbone in phospholipid. The carboxyl group in fatty acids were basically the components that help in the uh, the hydrophilic nature of the fatty of nature of lipids, while the other hydrophobic components were just the carbon skeletal chain. Then we go to question 6. Question 6 says, describe the biochemical basis of the use of ALP and ALC's liver function markers. So one important thing we need to know is that these enzymes, ALP and AST, aspartate aminotransferase and aminotransferase, are enzymes that are very much active or being expressed highly in the liver to carry out a lot of metabolic activities. So they are expected to be there highly. But any situations that we want them to be found inside the blood or their concentration exceeded expected to be seen inside the blood, it simply says that there is a problem with liver, and classically is an indicator of liver damage. So this is basically the reason why it is being used. Now the next thing, the question 6b says explain non-competitive inhibition. So non-competitive inhibition occur when the compound or a molecule binds to the aside from the active side of the protein. So it does two things, or it inhibits a protein in two ways. Either it inhibits a protein by inhibiting the enzyme substrate complex, or it inhibits the enzyme. So if it inhibits or it binds to the site before the binding of the substrate, it can lead to conformational changes of the protein, which also bind, but it may not be able to be converted to a product. Or even after the substrate already binds to the active site of the enzyme, but this inhibitor can bind to its own binding pocket, thereby preventing the conversion or separation between the enzyme and the substrate. Because if the enzyme is already attached to the binding pocket, and what happened leading to conversion of the substrate to product. The binding of this inhibitor may prevent the release of this product. That is what we call non-competitive inhibition. The question 6C says define synthetic nucleotides and state their use. So we say synthetic nucleotides are nucleotides that are artificially produced. They are analog to the nucleotides you mentioned, the basic ones we see in DNA and RNA. But remember, we said this structure, the purine and pyrimides, they are so diverse. So of course, we can have some other purines and pyrimides that can react with the ribose sugar and the phosphate group to form a synthetic nucleotides that look exactly like the nucleotides we know, the 6 nucleotides. And they have been used. But the normal nucleotide can be synthesized and used for DNA sequencing for PCR. But some nucleotides that are analogs, they are different from the normal ones that we know, has been used in drug development, where we give an example of anti-cancer agents. We made a mention of quite a number of anti-cancer drugs that mimic the properties of this nucleotide and ended up inhibiting some of the enzymes like PLA polymerase and others. We also made a mention in the case of antiviral compounds like in HIV/AIDS, where we have some of these compounds mimicking the structure of those nucleotides and ended up in preventing or inhibiting like viral transcriptase or polymerase, etc., etc. So this is basically what you are expected to do. You expected to be brief in your answer, but brief, not too brief, not too brief. Try and capture the most important things you are expected to, because there are some people that will even start explaining the question. When you ask the question, they start from the scratch. They don't go direct to the answer. They start from the scratch, the explanation that leads to that. Or reaching the point where they will mention exactly what you expected to mention, then you just put a post stop. So I don't know. In that situation, actually, you didn't say what exactly you expected to say, but you said something that leads to that. But, so the most important thing is take your time, understand the question before you uh, know what exactly you were asked. So, like while saying, you can, it's a good thing to say something that leads to what you're supposed to say. So, it's good to say that, but make sure that you say what you expected to say. But if you choose to go straight to say what you expected to say, that is also fine, and it takes less time for you to deliver. So, I wish you all the best. I was unable to get all the questions, but at least I'm sure this one will help you a lot. So please take your time, revise this thing, look at it again, and practice all the questions. So you sit down and think. So when you read, you should think of this kind of questions and see if you can be able to answer these questions. It tells you when you come across similar questions in your examination hall. So I wish you all the best. Please, for those of you that are yet to submit their assignment, please try and do that. Both the individual assignment, I can see the individual assignment. There are some that are yet to submit, and for the group assignment, almost half or more than half of the group are yet to submit. Please try and do that. I didn't put the deadline that I will soon put, and the time I will put is when that is 2 days to the deadline. I'll just set the deadline and then I'll set it. So if you fail to submit before that time, please, it simply says that you have not been working on it. So please do that. And for those of you also that have not done the test, I think the test will start tomorrow or next tomorrow for your information. So make sure at least you are able to. The last thing I want to say is this class or this tutorial, I've given an AI reading of it, which was the one you were initially watching. So it is there. Also, you can listen at that one. You can listen at this one. I've also created a revised revision of all our classes and uploaded on this YouTube channel. So I'll also tell you about it, and you go there and also revise so that you can just sit down and listen to it while maybe while you're about to sleep, you can just be listening smoothly, because it's AI dimension, all those things, and will capture definitely what you expected to learn throughout, and then make sure that you learn what you expected to learn, so that you later, not even for the exam set, you should be able to, whenever biochemistry is mentioned, you should start thinking of the chemistry that occurs in the biological system within your brain without saying, I have to take my note, I have to take this, things like that. So, having said that, if you have any question, if you have anything regarding the just discussion we have today, please you refer to the WhatsApp group and drop your comments, and then I will respond to you immediately. I wish you all the best. Bye. Assalamu alaikum. Assalamu alaikum. Allah. Me like, oh, okay. Masha Allah. Masha Allah. Biochemistry, Dr. Aminu Ibrahim. Dr. Aminu Ibrahim. Yes, yes, sir. Yes, sir. Okay, akwai dai, yes, yes, ba nan dai ba, akwai dai former HO din su, wani Prof. YY Muhammad, Prof. Yusuf, Prof. Yusuf Muhammad. Yes, so, okay, daman da shi muke ta communicating sosai dama tun can baya. Yes, sir. Dr. Aminu Ibrahim. Yes, sir. Oh, oh, okay, sir. Yes, sir. Oh, okay, sir. Yes, sir. Yes, sir. Oh, okay, sir. Okay, okay, I see. Yes, 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 sir. Yes, sir. Na fahimta. Eh, na fahimta. Yes, yes. Na gane. Ayya, ayya. Ikon Allah haka ne. Haka ne, exactly. 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 To, na gode sosai, sir. Na gode sosai. Zan nemi ka zuwa jima, insha Allah. Amin, amin. Masha Allah. Thank you, sir.